we examine the most substantive issues arising from critical scientific material.
The focus today is on the promise and the inherent limitations of manipulating the epigenome. That's the intricate system of modifications, things like DNA methylation and histone changes, that controls our gene expression without actually altering the DNA sequence itself.
Right. And the central question is this. Given that the epigenome is so dynamic and interconnected, can pharmaceutical strategies reliably achieve truly targeted, specific therapeutic effects? Or are we, fundamentally limited by the system's complex, adaptive nature?
Exactly. So my position is that these epigenetic mechanisms represent valid, druggable targets, and that increasing our molecular specificity is an achievable and, I think, necessary therapeutic goal.
And I'm going to argue that the non-independent, dynamic, and environmentally responsive nature of the epigenome fundamentally limits the successful application of targeted therapy. I think any sustained clinical success is going to remain contingent on these broader, non-specific systemic treatments.
Okay, so let me lay out my case. Epigenetic processes are, they're now irrefutably established as drivers of numerous diseases, especially in cancers and neuropsychiatric disorders. The system isn't abstract. It's driven by tangible molecular machinery, the so-called writers, erasers, and readers. The existence of enzymes like DNA methyltransferases, or DNMTs, and histone deacetylases, HDACs, provides discrete, known chemical reactions that we can inhibit or promote. We already possess proof of concept and, frankly, therapeutic success. You can just cite the FDA approval of 2 HDAC inhibitors, vorinostat and romidepsin, for cutaneous T-cell lymphoma. That demonstrates clear clinical viability. And more importantly, when we really dissect the mechanism, we see that precision is possible. Take valproic acid, VPA. It's known as an anti-epileptic and a mood stabilizer, but mechanistically, it acts as a potent direct inhibitor of class 1 and class 2 HDACs. Inhibiting these specific classes leads to increased acetylation on histones H3 and H4, which in turn promotes the expression of key neurotrophic genes like BDNF and GAD. This isn't some random side effect. It's direct molecular manipulation leading to a therapeutic outcome. And beyond that, we are actively moving past this kind of broad inhibition. Next-generation research is identifying compounds with true selectivity. We now have inhibitors like UNC0638, UNC0646, and UNC0631, which selectively target very specific protein lysine methyltransferases like G9A-GLP. This work in methyltransferases, a highly complex class of enzymes, is the very definition of achieving molecular specificity. It confirms that the limitation is in our current technology, not in the principle itself.
Okay, but I come at it from a fundamentally different way. While you focus on the potential of these individual targets, I'm focused on the holistic nature of the regulatory network they live in. The epigenome is not a series of independent light switches. It's a profoundly complex, interwoven regulatory circuit. It's got DNA methylation, a huge variety of histone modifications, acetylation, phosphorylation, ubiquitination, and non-coding RNAs. The crucial issue here is that these components absolutely do not act in isolation.
Sure, I agree with that.
And this interconnectedness is precisely why non-specificity remains such an intractable risk. Look at the current DNMT inhibitors. Non-specificity is a major concern because broad demethylation can lead to the activation of previously silenced genes, and that often dangerously includes oncogenes, right alongside the desired therapeutic effect. This non-specificity forces compromises on dosage and duration, which results in systemic toxicity. And even when we try to target a seemingly defined group like the HDACs you mentioned, the targeting is complicated. We classify them into subclasses like class 1 and 2 because they have distinct roles, but our understanding of the differential mechanism of action among these various subclasses is still heavily debated even among experts. So inhibiting class 1 and 2 HDACs globally is powerful, yes, but it risks disrupting myriad cellular pathways all over the body, not just the diseased one. how can we really call VPA specific when it causes such wide systemic changes? And finally, that's not even considering the external factor. How can we establish reliable long-term dosing when the target itself is constantly being redefined by things like stress, or diet, or inflammation? This continuous acquired dynamism fundamentally defeats the stability that you need for predictable drug targeting.
While that study does highlight the necessity of robust diagnostics, I think we might be misinterpreting the clinical significance of that heterogeneity. We have to refine clinical use based on this observed variability, rather than letting that variance just halt all the research. I mean, consider the classic azacitidine study in myeloid leukemia cell lines, the HL60, K56, and HEL lines. Yes, it revealed highly heterogeneous demethylation patterns, even under carefully controlled equitoxic conditions. HL60 cells showed substantial demethylation, K52 showed a minor but detectable change, and HEL showed none. This heterogeneity, to me, mandates better patient stratification. It doesn't negate the drug's mechanism or its potential. The fact is, the drug worked precisely as intended in the HL60 cells. This heterogeneity is cell line specific. It's not evidence that the epigenetic principle is flawed. Array-based analysis confirmed that in the responsive cell lines, the drug achieved demethylation of numerous specific hypermethylated markers. So the biological potential, the capacity to hit the target and reverse these aberrant marks, is proven. The clinical challenge is simply identifying the responsive tumor environment, which is a problem of patient genomics and companion diagnostics. The solution isn't giving up on the target, but refining our ability to match the mechanism to the context.
Okay, that's a strong mechanistic anchor. I will concede that the HL60 data clearly demonstrates the mechanism can be effective. But I would argue that this extreme heterogeneity is the central problem for therapeutic translation. We're relying on assays that are themselves inconsistent, which really erodes the foundation of predictability that you need for clinical application.
How so?
Well, let's focus on those K562 cells you mentioned. In those cells, capillary electrophoresis, which is a generic assay measuring total genomic cytosine methylation, it measured a significant overall reduction. Yet, at the same time, gene-specific methylation assays, which look at the methylation status of known tumor suppressor promoters, failed entirely to detect any changes in those specific genes. So, if the diagnostic tools and the cellular responses vary that dramatically, where the global measure says yes, but the clinically relevant measure says no, how can we possibly assure predictable outcomes in vivo? The systemic toxicity and off-target effects of DNMT inhibitors remain significant precisely because we lack this predictable outcome. If success relies on finding a small, perfectly responsive subpopulation like the HL60 cells, and just accepting that the drug is completely ineffective or dangerously broad everywhere else, then the targeted approach is inherently limited. The therapeutic goal is systemic disease management. And if the epigenetic state is so highly contextual that it makes predictability impossible across patient heterogeneity, then the drug class has failed the specificity test.
I think we need to elevate the discussion here beyond simply inhibiting known enzymes and look at where the fundamental regulation actually originates. Epigenetic regulation is fundamentally and directly tied to cellular metabolism. And this relationship provides critical sophisticated intervention points that might bypass this issue of systemic complexity that you're raising.
Okay.
This is basic biochemistry. Nutrients involved in one-carbon metabolism, specifically folate and B vitamins, they regulate SAM synthesis, and SAM is the required universal methyl donor for DNMTs and histone methyltransferases. Similarly, acetyl-CoA, which is produced directly by the TCA cycle, dictates the available supply for histone acetylation. When the cell is metabolically robust, acetyl-CoA is plentiful, and transcription-activating histone acetylation increases. This directly links the cell's energetic state to its gene expression profile, and this link allows for a form of highly sophisticated targeting, modulating these metabolic pathways chemically. We already see this in nature. Polyphenols, for instance, they modulate epigenetic patterns by affecting DNMTs and SIRT-twin activity. If we can leverage natural compounds that affect these metabolic inputs,
we can certainly design synthetic drugs that modulate the production or utilization of SAM or acetyl-CoA with far greater selectivity. This demonstrates that the epigenetic machinery is an elegant metabolic sensor, and it offers a robust pathway for intervention that is, upstream of the enzymes themselves.
I agree that the metabolic input is critical, but are we not just swapping one layer of systemic complexity for another when we propose targeting SAM synthesis or acetyl-CoA globally? Acetyl-CoA is fundamental to virtually all central metabolic processes, and SAM is used in hundreds of transmethylation reactions beyond just DNA or histones. Targeting these bottlenecks globally guarantees off-target systemic effects that could, frankly, dwarf those caused by a simple DNMT inhibitor.
Well, it requires a different kind of specificity.
Perhaps. But furthermore, the evidence from neuropharmacology really challenges the whole premise of direct targeting. Many successful psychiatric drugs achieve their epigenetic effects entirely indirectly. When we prescribe an SSRI or a psychostimulant, the primary target is the monoamine transporter or receptor. That primary action then triggers a complex signaling cascade via transcription factors like FOSB or CREB, or it leads to a secondary reduced expression of DNMTs in HDACs. The critical point here is the mechanism of action. We are designing a drug to hit a single receptor, and then we observe a downstream epigenetic consequence that's 10 steps removed. The lack of clarity on how that initial blockade translates into specific, lasting brain epigenetic effects really proves the system resists direct, specific manipulation. The epigenetic changes we see are often just a secondary, often uncontrollable consequence of a non-epigenetic drug action. If the body finds it easier to use an indirect signaling cascade to get an epigenetic change, why should we believe our direct enzymatic targeting will be stable and reliable long-term?
The distinction, though, between direct and indirect is blurring as we gain more mechanistic insight. And that blurring is what opens new avenues for multi-target intervention. Progress is reliant on identifying and mapping these complex interactions, not treating them as insurmountable barriers. And we are making significant gains in understanding this crosstalk between different epigenetic marks.
Okay, but it's still early days.
It is, but look at the studies investigating it. For example, in studies involving KDM6A CKO sperm, we observed a clear regulatory interplay. KDM6A is an enzyme that removes H3K27Me3, which is a repressive histone mark. When KDM6A was conditionally knocked out, the regions where H3K27Me3 was perturbed, either gained or lost, were consistently associated with increased DNA methylation in those exact same regions. This is not just random noise. It demonstrates a defined, predictable regulatory relationship where repressive marks coordinate. This knowledge allows us to design combination therapies that target both marks synergistically, maximizing precision. Moreover, this complexity is evidence of a plasticity that we can leverage. Physical activity, for example, induces an epigenetic memory in skeletal muscle, which enhances metabolic adaptations long after the exercise is over. This shows the epigenome is capable of encoding these long-term adaptive functional states based on environmental input. If we can reverse engineer this beneficial biological programming pharmacologically, we are essentially harnessing the system's own adaptive capacity for therapeutic gain.
I'm just not convinced that simply mapping crosstalk solves the problem of predicting long-term outcomes in a living patient. That detailed mechanistic work, while it's crucial, it still remains perilously reductionist. Most studies, especially those designed to test a single drug, they focus on isolated mechanisms. They observe only DNA methylation or only histone acetylation.
Well, you have to isolate variables to study them.
Of course, but the major systemic limitation we face now is the persistent lack of integrated multi-omics approaches in long-term clinical studies. We can map the H3K27M3 and DNA methylation crosstalk in an isolated model, but without integrating the full cascade. The histone changes, the methylation, gene expression, metabolomics, over sustained periods in vivo, we will struggle profoundly to disentangle which epigenetic changes are truly causative of the disease reversal and which are merely compensatory or reactive changes to the drug itself. If we try to reverse engineer this beneficial plasticity, we have to account for the fact that the system's adaptability is a double-edged sword. If the system is so adaptive, it will constantly try to neutralize or compensate for the pharmaceutical intervention, and that leads to drug resistance and the failure of long-term therapy. Until we have that full, comprehensive systems view from integrated multiomics, our ability to design truly effective, long-term epigenetic therapy for systemic diseases will remain constrained by, well, overwhelming uncertainty and high risk.
The challenge you present, this necessity of systems thinking, is one we absolutely must meet. But it doesn't diminish the validity of the molecular targets themselves. We have to remain committed to refining specific drug strategies, moving beyond the current limitations of these first-generation inhibitors. The epigenome is highly dynamic, yes, but this dynamism is what makes it plastic and therapeutically responsive. Our path forward is clear. We must continue to focus on the molecular machinery, the DNMTs, HDACs, HMTs, while simultaneously leveraging insights from systems biology. By understanding how lifestyle interventions like diet and exercise naturally modulate metabolic inputs like SAM and acetyl-CoA, we can design next-generation inhibitors that are not only more potent, but far more selective, potentially through combination therapies that target this regulatory crosstalk. The success of targeted drug development in other complex biological systems assures me that this level of specificity is within our reach.
That's a compelling argument for the future of research. I will grant you that. But I have to return to the idea that the fundamental difficulty lies in the interconnected, global nature of these epigenetic marks and the sheer heterogeneity of the cellular response. The inherent complexity of chromatin architecture and the profound influence of the external environment on these acquired epigenetic states suggests that true targeted precision remains an ideal rather than a reliable clinical reality for systemic disease. For long-term clinical success, I believe we must prioritize understanding the integrated systems biology to fully predict and mitigate the systemic consequences of our interventions before we can reliably deploy these drugs broadly.
I think this discussion really illustrates that while the epigenetic landscape holds vast therapeutic potential, the path to clinical success requires reconciling our pharmacological precision with the system's remarkable complexity and adaptability.
Indeed,